Hello, everyone. I have the privilege of talking about a number of people's research today, and some of them are in the room, so if you ask me hard questions later, I can point at that person. But I'm going to try to take you through descriptions of what they've been doing and let you think about solutions that are further away from us, perhaps, than fungicide use. Jim did a beautiful job of telling you where we are in terms of understanding fungicidal control, and 95% is pretty good, so that's not so bad. But we're trying to also, as a group of boxwood researchers, look into the future, think about neat, interesting new approaches that might allow us to use newer technologies to get us somewhere with control of the disease. So this is our enemy, the Calanectria pseudoniviculata. It has large, sticky spores, and it is what is causing the infection of the boxwood plant that creates disease symptoms. And I just want you to realize that it's not the only thing there. We tend to simplify as we think about these and get the idea that, okay, you add a fungus to a plant, you get an infected plant. Well, there's a lot more going on there and many more microbes present than what you might think at first. And I'm sure they all look the same to you. But this is one example of what we see in a diagnostic lab. When you get a brown or partially brown boxwood leaf, the landscaper or arborist or grower wants to know if it's boxwood blight. And you incubate it under humid conditions to get sporulation so you can answer the question. And in this example, well, there's a little patch over here that actually is the Calanectria pseudoniviculata, but it probably looks a lot like the stuff over here to you. And actually, here's another patch of the Calanectria, but I think this is probably a fusarium, and this whole section through here is Volutella, which looks exactly like it until it matures a bit. It gets pink eventually, but it starts out white, and everybody who's done this knows exactly what I'm talking about. So we do see, if we're working with these plants in the lab, that there's a lot more biology there, but we're still just looking at the tip of an iceberg. There is a great deal of microbiology present in this system that we're talking about, and one of the ways we'd like to approach control is to learn ways that we can harness that microbiology for good purposes. And so I'd like you to think a little bit about the microbiome, which is a fancy word for all the microorganisms that are associated with a plant. And one of the concepts here is the idea of an endophyte. So endophytes are microorganisms, usually fungi or bacteria, that are inside the plant tissue. But they're not creating any symptoms. When we have something inside the plant that's causing a symptom, we think of that as a disease problem. The situation that you have there is one that's sort of a mutualism. The plant is providing carbohydrates for the benefit of these endophytes, and the endophytes themselves, we're beginning to learn more about, are providing often a defense capacity to the plant. So there are benefits to having certain microbes there, similarly to what goes on in your own intestines, but I won't try to go there too far. When you have this symbiotic relationship, it's not always permanent. So there are some organisms that can be beneficial, living in harmony for a while, but then if the situation changes, they may turn mean and start attacking the plant. And there are other situations where they just protect the plant. So here's an example of one of those more complex interactions, but it's one that Chuan mentioned and others have earlier today. This is the Volutella blight, the other boxwood blight, which was in existence for many, many years before we started worrying about this new one. And Volutella will cause death of leaves, it will cause death of shoots, but it needs a trigger for that to happen. It's not able to force its way into the plant. It needs a wound. It may need a stress. So we think about Volutella going after frost-injured plants or plants perhaps that have been injured by a boxwood leaf miner or something like that that creates a physical injury. Those kinds of things, including all of the shearing that we do. That provides an opportunity for Volutella. So it's a wonder, actually, that we can deal with it as calmly as we can. It could be really rough considering how we treat the boxwood sometimes. But I'd like you to think about potential for it to do something in the future. This is an example of an isolation plant from the lab where we've looked at pieces of tissue that are showing symptoms that we think might be boxwood blight. And we have actually succeeded in isolating the Calanectria in several cases. You can see these large brown colonies. Those are typical Calanectria. There's a bit of it here. There's even a tiny smidge of it here. but this large white growth with the orangey sporulation is Volutella. And we see it all the time. It's virtually always there, and it's waiting for its chance to perhaps attack the plant when there's a stress. But I suggest to you that just perhaps we might be able to make use of it, because if we were to alter Volutella or discover a Volutella that had this tremendous ability to be an endophyte, which is exactly what it is, it's in the plant at all times, we might be able to use it as our way to deliver some extra defense capacity to the plant. So we've already got right in front of us something which with a little bit of change might be very beneficial, and perhaps we just need to discover a strain that has no negative effects on the plant that does have this great ability to be an endophyte. So that's one concept for the future for you. And another thing that is an interesting idea is the idea that we could go to our own Boxaceae native, which is the Pachysandra that's the procumbens, which is the Allegheny, used to be Allegheniensis, the Allegheny Pachysandra, and look to see what is in its microbiome, maybe on the leaves of that plant or in the soil surrounding that plant. There are some things that are beneficial for that kind of family. The idea here is that maybe as we have adopted and domesticated the boxwood plant, we've left some of its beneficial organisms behind. And we happen to have one American native on the continent that we could go to and use to check out some interesting possibilities of finding microorganisms that could help us fight back against boxwood blight. There is a project similar to this that Joanne Crouch and her associates have been involved in to look at the microbiome of the boxwood plants at the National Arboretum and also at the State Arboretum of Virginia. And they have done a tremendous job. When you see work that's done in this area, it's always so impressive because it's always thousands. To do this right, you don't go isolate 4 things from the plant. You look at 16,000 isolates, okay, in this situation, and you look to see what's there. You can now analyze DNA in masses as collected from the soil and so forth. So they have found some very interesting things in their first couple of rounds of testing along these lines. In particular, you see 4 of the trichoderma isolates that they have found in these studies. The trichoderma in this case is the dark stuff from where you're sitting. It's actually green if you had better lighting. So you have an inoculation here of the Calanectria, and over here the inoculation with Trichoderma humedum, and all this green is the spore production of the trichoderma, and the Calanectria hasn't been able to grow very well at all. It's being outcompeted. And similarly, you used to have a Calanectria buried under the Trichoderma aspergilloides there, and the Trichoderma tomentosum is really taking over here, and the Harzianum is taking over Trichoderma there. Taking over the Calanectria there. So we're seeing some obvious ability in the microbes that are associated with boxwoods that are growing even in our gardens. So as we sort through all this and sift through it, we may find an organism that can help us, much as now we are looking to chemicals to help us fight back against the disease. Here's some of Chuan Hong's work. He has been doing some of the first, well, certainly the first published information on biological control on the boxwood pathogen. And this is from one of his experiments. He worked with the Justin Browers, which you probably all know is a very susceptible cultivar that was mistakenly identified as being a Korean box at first, and now we know it's really just a sempervirens. And he did inoculations of the beneficials that he was testing 2 weeks before he did his trial, and also just a few hours before he inoculated them with the Calanectria. And the way you do this kind of thing is to collect conidia in little liquid solutions and spray them onto the plant. And it gets confusing because you're inoculating and then you're inoculating because you're using microbes for both stages. But at the end of it all, you can see whether your disease success is greater or less. And here's some of his data showing you the benefits of some of these treatments. Over here at the left, you can see the names of the materials. This includes some that are actually registered materials. The plant helper is a trichoderma material. The companion is not a fungus, but actually bacteria, that's Bacillus subtilis. And Root Shield Plus is a combination of 2 fungi what used to be called a gliocladium, now Trichoderma virens, along with the Trichoderma harzianum. And then down here is daconil, which is familiar to you as a competent chemical control for the disease. The way these were rated was to look at something called the area under the disease progress curve, which is a fancy term for how much disease there is. And if you look through here, if you did not treat the plant, but you inoculated it with the pathogen, you've got an area of 62. And then let's see, look down here, 34. With the root shield plus treatment, you're seeing what is almost 50% reduction of the problem. So that one really is promising. The others are not really statistically better, but this is a handy, available, we could get this disease on the label if we ask nicely, kind of a possibility. So this is one of our first indications that we might be able to get somewhere with biocontrol on this disease. Another experiment that Chuan did recently was to work with Trichoderma conningitis. Whoa, it almost went out on me. And that was, I think, isolated from a mushroom. Just nod if I'm right. It's a one that has not been used before in commercial products, but it's got some possibilities. And in this image, you can see 3 different isolates of the Calanectria, the brown fungus here, trying to grow on the plate. And in the presence of that particular new Trichoderma conningiopsis isolate, the bars will show you there's a great deal of reduction of growth, and you can see the colony is very tiny in that plate when the biocontrol is added. Here's another case of that. It's really good to see that happening in the lab, and you hope that the same kind of thing transfers when you take the organism outside and ask it to do a real life situation. And here's sort of a growth chamber greenhouse kind of trial showing you something along those lines. Here you've got plants that were looked at 5 days after the inoculation took place with the pathogen, and the ones that were treated with the isolate of trichoderma are looking pretty good here, and you're already starting to see little leaf spots showing up caused by the Calanectria where there was no biocontrol present. And down here on the Justin Browers, you have plants that are looking relatively healthy and ones that are starting to show a lot of leaf symptoms when they were not protected by the biocontrol. So this is just the beginning, I hope, of a wonderful series of trials that will perhaps give us something that will be helpful. I know from my own perspective, people ask me for an organic solution to Boxwood Blight all the time, and my answer is usually Ilex. But I think if we had some other treatments, it would make a lot of people very happy. Don't think you can do it tomorrow, but we're working on it. This is one of those new technologies. We did find in Chuan's experimentation that he had less disease develop if he gave the system more time to interact. So with 18 days between his inoculation with the biocontrol, when there was 18 days, he was able to get better effect than if he only gave the system 9 days before he challenged it with the pathogen. And these are all the kind of details that the experimenters have to work out for you. Another whole sphere relates to the sanitation that we talked about earlier today. And this is some of Nina Shishkov's work. She just gets to introduce us today, but she works real hard most of the time. And this started out at Fort Detrick, where she works, which is formerly known as the Foreign Disease Weed Science Research Unit's home. And this allows containment, so she can work with really very dangerous things under lock and key. And at that facility, they used to have a cute little greenhouse with lots of plants inside it, but they decided to completely destroy it so that they could ask these questions. And so they set up steam beds in that greenhouse to find out what happens with heat. Because if you were a nurseryman involved in propagation of boxwood, you would be really upset if you had this disease come into your facility, and you would absolutely want to be able to get rid of it completely. So this relates to our ability to eradicate. I had to have extra coaching on this one because I thought a little rope like this would be tricky to get things inside, but I was told it was actually leash material, and you can insert little tiny cuttings of boxwood into it and then bury them into a situation where you're applying steam and check to see if you're able to do anything about the fungus that is infecting those little cuttings. So this is a really scary table of data. But I just wanted to mention to you that it was really thorough. They did about 3 controls, whereas most of us would just use 1. and really compared what would happen if you did nothing to what would happen if you had temperatures between 150 Fahrenheit and 210 Fahrenheit, which would be considered a steaming treatment to get rid of something biological that you didn't like. And if you can see over here at the far side, they were measuring how many infected, what the percent of infected leaves was, or what the percent of infected twigs was. And at lower temperatures, hey, there was still infection, but there's a lot of zeros here when you get in that 150 to 210 Fahrenheit category of a successful steam treatment. So if you can get steam to this organism for at least, well, this was 6 hours at that peak temperature, you can eliminate this organism. It's just not usually convenient to get steam everywhere you'd like to get rid of this organism. Okay. And here's the visual proof of what I was just describing. Here's a typical leaf that looks sort of miserable because it's completely been invaded by the fungus, covered with the white sporulation of the Calanectria. Here's a little twig section that's also showing little sporodocia of the pathogen. But after the steaming, hey, it's all incapable of growing because it has been eliminated. The microsclerotia themselves have been eliminated, both on the twig tissue and the leaf tissue. So it's good to have the scientific proof of that ability. And then Mark Cabrera has been working alone and with Nina on coordinating information on heat treatment on boxwoods, because we don't always want to do something quite as thorough as complete sterilization. And he's looking at the benefit that heat treatment might have in propagation. So the first question is, what's it going to do to the spores of the Calanectria? Are you going to be able to kill them? And they are tricky things to kill. I just wanted to call your attention to this picture over here. We talk about spores germinating, and that usually means it's acting like a seed and something comes at the end and it goes on to grow. With this Calanectria pseudonaviculata, it has 2 spores. They both are capable of growing. So you're looking here at the 2 germ tubes. It's got 2 chances to start with. All right, a very hard thing to really inhibit. In Mark's experiments, he looked at temperatures ranging from 113 Fahrenheit to 131 Fahrenheit, and he looked at a range of different isolates taken from English boxwood or plain strain sempervirens, and had a really nifty system for doing that. He would collect spores, and he would put them into solution and then insert them into a thermal cycler that's ordinarily used for DNA studies, and treat them at a particular temperature for a particular amount of time, plate that out on agar to get the growth, and then examine whether or not spores had germinated, which means looking at those 2-celled spores and seeing if you've got something coming out of one end or the other. So you can really tell whether or not the heat has caused the spore to be completely inactivated or not. And not too surprisingly, his hypothesis was fulfilled. There was worse effect on the spores the higher the temperature went. Here at 113, these are isolates from a collection of different isolates. They don't all behave exactly the same, but over time as you raise the temperature, you can see it's harder and harder for those plants to survive. So you're killing 90% of the conidia at 8 to 16 minutes, for example, at 113 degrees. Which is hopefully enough, because that actually seems to be the best tolerated, as you'll see in a few minutes. But just notice the variability. It is important for us to not just work with one isolate and assume we have the whole story just with the one. And so now he's interested in combining the data of the effects on the spores themselves with data on what happens to the roots, because you're going to really want to be able to have good plant tolerance at the same time that you have very poor tolerance by the Calanectria. And the assumption is that rooting might be a bit disturbed by the high temperatures that would kill spores. So he's set up some experiments to start working in this area, and is trying to use a range of the boxwood in the trade that would be relevant to this question: green velvet and Nana and green beauty and D-Runk and fastigiata and Safruticosa, which includes a lot of Asian types and one European type. And he's got a rooting study experimental design that involves using little tubes that have holes in the sides. He puts his cutting inside that tube, puts them in a water bath, and he can apply the temperature to that cutting, and then afterwards compare the rooting ability of one cutting to another one that perhaps was in that temperature of water for 5 minutes more, or 10 minutes more, or 15 minutes more, just to get a handle on the plant's ability to put up with these heat treatments. Then he grows them out for a 91-day period and sees what he gets. And he's looking for decent rooting, and you all probably have opinions on this in the room, but he was separating the sheep and the goats by deciding that if he didn't give it a 3, 4, or a 5 rating, which represents these 3 here, it was really too poor to think of as a rooted boxwood cutting. So he was really looking for reasonably good numbers of roots in the plants. And you could see above ground some effects with these treatments. Going from left to right, you have higher temperatures on Fastigiata, and you can see there's an increase in the amount of browning in those plants. So if there's browning above, you know there's bad effects below as well. Here's an example of the information comparing 113 Fahrenheit to 122 Fahrenheit, and you see more bars at the left than at the right, because at that 122 degree temperature, you really didn't get any rooting at all out of some of the plants. And you can also get a quick sense for some of the cultivars being really different from others. Your Green Velvet and the Nana and the Green Beauty are pretty tolerant. You know, you're getting reasonably good survival of those, and good rooting in particular, even at 113 degrees. Whereas, let's see, this is D-Runk, not doing too well, Fastigiata and Sofruticosa, harmed by the 113 and having a much worse problem at 122, just not able to root at all. And here he's showing bars, giving you an idea for how you might set standards in that situation. You'd probably work with no more than 15 minutes for your treatment if you wanted to have the majority of your cultivars surviving. And if you cared about one of the others a lot, you might have to adjust that as well. When looking at this, the cultivar variation was very important to him, so he developed information on that. The one which stuck out in this first group was Nana. It was not as tolerant as the others. It was often the only one that was statistically different in terms of its survival after the temperature treatments. And when you increase the time or increase the temperature, it really stood out. The different colors here are showing you different cultivars. So you've got the Justin Browers here. Yes, there's a reduction as you increase the time in the heat treatment. The last point here is actually room temperature, so don't look at the last point. But you get with each one of these here with your Nanas, I guess, no, excuse me, the Nana's the red bar that comes really plummeting down low. That's the one that's really statistically different from all the others, has a very poor ability to survive these heat treatments. And don't try to do anything more than get just a general sense of this. I know some of you are 50 yards away, but I think if you just look at how much is showing in those colored bars, you can get a sense that if you use a lower temperature, here's 113 Fahrenheit that was used on different cultivars, each clump here is a cultivar, that you will have not all that much inhibition of rooting. There is a reasonable amount of rooting going on here, but if you increase the length of time they're treated at that temperature of 113, there's a lot less rooting. And if you increase the temperature itself, you also have less rooting than you see here. And the overall pattern is that the ones that come from Asia, which perhaps are normally subjected to warmer temperatures, are having less trouble with this heat treatment idea than others. So perhaps this would be something you could use in propagation with certain of your cultivars, but you might not want to ever touch it with English boxwood. My sense with that is that it never wants to root when I looked at his data. So in this case, his Asiatics were Green Velvet and Nana and Green Beauty as compared to the Sofruticosis. Here you're looking at leaf damage a month after treatment, just to give you another sense that there is some above ground visibility here too. With your Green Beauty treatments, the lower treatment at lower temperature treatment of 113 gave you the healthiest looking plants. And so he's going to be moving from these kinds of studies into seeing what happens when you start with an infected cutting and you're treating it and you're trying to see, gee, does it really clean it up during the propagation process? That would be truly marvelous if we could get rid of some of our contamination through these kinds of procedures. And there's one other little side thing he's noticed, which I think is very interesting. There are times when your spores aren't able to germinate, but at that treatment, even if you've gotten the germination down to really low levels, let's say 3 to 8%, as you see there, there might be no disease. You may not need to get it down to 0 germination. So there are impacts on the spore's ability to create disease that are greater than just germination. So we can't just do lab tests here. We're going to have to actually work with real plants to see what's going on. And in the course of things, he also got the sense that there were times when you could treat with enough heat that it almost made things easier for the pathogen, because if you get it hot enough, it seems like you select for the heat-tolerant individuals in the situation. So we have to learn more about what we're doing there and maybe use 2 methods at one time so that we don't just increase the heat tolerance of Calinectria. One final thing is that we're starting to learn other ways of detecting this pathogen. It's a lot of trouble to do PCRs in the field. So Doug Luster and Joanne Crouch and Nina Shishkoff have been working together on other kinds of methodology using serology. Some of you may be familiar with the test strips, immunostrips that Agdia sells that help us identify viruses and some bacteria and some oomycetes for that matter. And they're looking for similar kinds of things. They're going to get the immunoassays going by coming up with new techniques for detection of this based on an immune response. So first they have to develop both polyclonal and monoclonal antibodies to work with, and then ultimately they're hoping to have both ELISA-type laboratory tests and also immunostrip assays that would be what you could do in the field if you happen to be a plant breeder trying to screen 10,000 plants in the field, for example. And that will really help landscape gardeners and plant breeders to make at least an initial evaluation. You just can't always go to a plant pathology lab when you want to know what's going on.